Picking up with our conversation on proteins, we need to look at those 4 levels of protein structure. Some instructors go into a really deep format here and they dive down hard into this, and I tend to not. I tend to look at them by definition and then move on from there, because what I really want us to do is to gather some information about really the function of the proteins. Remember that the function will be a result of this structure. So a primary structure of a protein is its unique sequence of amino acids. Secondary structure, which we find in most proteins, consists of coils and folds within the peptide chain. Tertiary, I said that poorly. Tertiary structure is determined by interactions among various side chains, or R groups. So those side chains can actually start to interact with one another. You know, once you bend something back or coil it back on itself, now you put those side chains in close proximity, and we'll start to see a different structure based on the interactions between those R groups, those side chain groups. Quaternary structure results when a protein actually consists of multiple polypeptide chains coming together. Very similar to what we saw when we just looked at that antibody and that virus. So that would be a quaternary structure sort of situation. Here's an example of a change in a primary structure. So this is, we're going to talk a little bit about sickle cell anemia here, or sickle cell disease. And that is a change in the primary structure. It's a very slight change. It's a single amino acid substitution. So what ends up happening is the structure of the protein is altered by simply replacing out one amino acid with a different amino acid. So it's sickle cell, if you're not aware, is an inherited blood disorder. We have a single amino acid substitution. We talk about sickle cell specifically later in the semester when we talk about transcription and translation and protein synthesis. So I'll save the big reveal until we get to there. But in these 2 images here, down on the bottom of your slide, you can see on the left a normal red blood cell. Normal red blood cells are full of an individual hemoglobin molecule, each carrying oxygen. What we see structurally, physically, in that red blood cell, what we see is a biconcave disc. And on the right, what we see is a sickle cell. So that is a red blood cell in the sickle shape. That's how the disease process got its name. And what happens is I have abnormal hemoglobin. So I have fibers of abnormal hemoglobin, and they actually are, because of their structure, they form differently, and they appear to be a deformed red blood cell. And here's exactly how, I guess I am going to do my big reveal. This is exactly how that has. So we have this primary structure going along here. You'll notice at number 6 on the left you have glutamate, and over on the sickle cell side, that glutamate gets replaced out with a valine. So what ends up happening is the secondary and tertiary structures are altered. You'll notice that difference between the left side and the right side of your slide. Because the tertiary structures are different, then that means that the quaternary structures have to be different, and we end up with a sickle cell shape. Function is very, very different between these molecules. Do not associate with one another, and each carries its own oxygen in the normal red blood cell. In a sickle, what ends up happening is those molecules do interact with one another, and they crystallize into a fiber. And that now crystallized fiber, while it has the capacity to carry oxygen still, it has it at a greatly reduced measure. You may or may not know that sickle cell actually is an advantageous trait in certain parts of the world. Parts of the world where malaria is common. The malarial parasite isn't able to survive on individuals with sickle cell shaped red blood cells. So those individuals have a genetic advantage over individuals with normal-shaped red blood cells who are then susceptible to malaria. Our next slide is what determines a protein structure. So in addition to the primary structure, we have physical and chemical conditions that are going to affect the structure. Alterations in pH, salt concentrations, temperature, other environmental factors can cause a protein to unravel or to fall apart. When a protein loses its original or what we call its native structure, that loss of the native structure, that process is called denaturation, or you'll hear that the protein has become denatured. It's not in its natural native state. A denatured protein becomes biologically inactive. It can no longer do its intended function. So again, we're going to talk about structure and function, and if you change the structure, you change the function. So you want to know that what denaturation is. Denaturation is, you want to know how the function of a denatured protein really is, and that is that it doesn't function its normal way. Can it still function in a different way? Sure, potentially, but not in its intended original method. So that makes a difference. We'll talk about, for those of you in lecture, face-to-face class with me, we'll talk about denaturing proteins in lab. We'll talk about acid and temperature and other ways to denature protein. For those of you in the online course, please just refer back to your textbook and read through that section extensively. Here's a great little whiteboard matching that I have set up for you. It's the last slide I have on proteins, and then I believe we move into nucleic acids. So there's a whiteboard matching. Go ahead and take a stab at that. Check your answers against the textbook if you need to. I'm going to wrap this one up. We're going to come back and talk about nucleic acids.